Okay, so we're going to look at the receiving systems that are stuck on the plasma membrane, and these would be for molecules that are too large or are hydrophilic and are not able to go through a cell membrane to get the signal to the inside of the cell. So basically the goal with these membrane located signal receivers is that they're trying to get the signal from the outside of the membrane, get it to the inside of the membrane where it can actually do something. And so this is where the chapter, I think, starts to get, I don't want to say confusing. It's just you could easily read an entire section, and when you're done, just be like, "What in the world did I just read?" Anyway, and it's because it gets pretty heavy on terminology at this point. So hopefully I can simplify it a little bit for you. I feel like I'm just saying what's in the book anyway, but hopefully I can simplify it a little bit. But anyway, the first type here is called G protein linked membrane receptors, and basically they are integral proteins that are on the membrane of the cell, and integral means that they go through the entire membrane. And there's a picture up here. It shows there's some alpha helices in there, which is good review from protein structure. But anyway, there's two locations on this that are of interest. There's one on the outside, which is where the signal, whatever signal is being received, where that signal is indeed received. And then there's a location on the inside that's going to interact with something called a G protein. And so we have this receptor here, and then our G protein. The G protein is a peripheral protein, which just means it stays on the outside edge of the plasma membrane, and it can move around there. So it can actually move around on the inside edge of the plasma membrane. And this G protein can either be active or inactive. The difference between the two is depending on which molecule it has on it. There's two options. It can have GDP or GTP. These two molecules are very similar to ADP and ATP. It's guanine diphosphate and guanine triphosphate. Very similar in structure. Anyway, if GDP, if that form of it happens to be on the G protein, it is not active. And if GTP is on there, then it is active. Those are two options. So once our G protein receptor, once it receives its signal, whatever that happens to be, then that activates the inside of the protein, that site on the inside, and makes it an active enzyme, and the reaction that it is going to facilitate is going to be the removal of the GDP, something we don't want, and we're going to place GTP on this G protein, and that activates it. So once that has happened, once that reaction has happened, then the activated G protein is just going to be released from the receptor protein, released, and remember it can move around in the plasma membrane. Actually, all these proteins can move around, but anyway, it'll move around until it reaches some form of an enzyme. And again, this is totally dependent on what reaction is needed and what response system we're set up on. But anyway, this G protein will find the particular enzyme that it can match up with, and now that it's activated, it will be recognized in the active site of the enzyme, and it will activate that enzyme. So once the GTP or the active G protein goes into the active site, then that enzyme can do whatever it's supposed to do, which is pretty broad statement there. But whatever it's supposed to do, we just activated it, and it can cause that to happen. Now, the G protein itself, once it has activated an enzyme, it will, most of the time, it'll go through the hydrolysis of GTP, where it'll knock off that phosphate that makes it TP, triphosphate at the end, and it will make it a GDP molecule, and so it will just have inactivated itself or deactivated itself, and so we're back to where we started. At this point, that G protein could be used once again to receive a signal from another activated receptor protein. So that's the G protein linked membrane receptors. That's how they work. Several steps there, but it's pretty simple. One molecule gets turned on, gives the signal to another molecule, which is then turned on, and then that goes and gives the signal to an enzyme, which is then turned on. Okay, so it's just turn on or activate a molecule, pass the activation onto another molecule, pass that activation onto another molecule is what we're seeing on this one right now. So that's G protein linked membrane receptors.